CRISPR evolved billions of years ago to protect bacteria from attacks by viruses. Over the past decade, scientists have determined how it does this and have harnessed its power to delete, edit, or replace plant and animal genes, including human genes. Today, I'll be discussing how CRISPR was discovered, how it is being used by plants and animals, how it is being used by humans, and its potential benefits and risks. In due time, I'll also tell you how CRISPR got its name. The story of CRISPR starts in the Mediterranean seaport of Santa Pola, located on Spain's Costa Blanca. The port is known for its beautiful salt marshes that have attracted vacationers and flamingos for centuries. About 30 years ago, a young Spaniard named Francisco Mojica, who we'll call Francisco, was beginning his doctoral studies at the university laboratory in Santa Pola. The laboratory was studying the bacteria found in the local marshes. More specifically, the lab was studying the bacteria's DNA. DNA is present in nearly all living organisms. It is like the string of genes. Different groups of genes on the string control different aspects of how an organism will grow and function. DNA and its genes are also the biological units of heredity and pass information from parent to offspring. A deep understanding of how DNA and genes work has become essential in agriculture, medicine, and many industrial fields. Under a microscope, DNA looks like two railroad tracks as shown in this graphic. The tracks are held together by sets of half ties called bases. Two bases that are connected to each other are called a base pair. Each set of base pairs that has a specific function like determining eye color is called a gene. The smallest human genes are 76 base pairs long, and the longest 280,000 pairs long. The latter gene plays an important role in muscle development and action. DNA is not flat as a railroad track. In fact, it's twisted. Here's an artist's stylized representation of what a twisted strand of DNA looks like. But let's return to Santa Pola. Soon after Francisco resumed his studies, he noted a curious pattern in the strings of base pairs he was examining. There are only 4 different base pairs in DNA, which I'll call A, B, C, and D. Most pairs are arranged in no obvious order. However, between each of these sets, he found sets of base pairs in palindromic order. That is, they read the same way forwards and backwards. You can see this pattern of base pairs in this drawing of a bacterium. Palindrome, spacer, palindrome, spacer, palindrome, etc. Francisco assumed the palindromes must be important. Why otherwise would Mother Nature go to the trouble of making them? And that the base pairs between them, as best he could tell, were just taking up space. So he named them spacers. Because the lab he was working in was so poorly funded, there was hardly any money for him to carry out lab experiments. So he decided instead to search online computer databases to see if this pattern of palindrome, spacer, palindrome had ever been reported before. He was surprised to rather quickly find reports of this pattern in the DNA of over 20 different bacteria, including tuberculosis and plague. Soon, as other researchers noted the pattern, it was given the name CRISPR, to stand for clusters of regularly interspaced short palindromic repeats. Theories abounded regarding the function of these palindromes, but one by one they proved wrong. So during his August holiday in 2003, Francisco gave up on trying to understand what they did and started to focus on what the spacers that separated them might be doing. He noticed that the DNA in the spacer that he was examining contained a set of 11 base pairs that exactly matched a set of 11 base pairs of a virus that was known to infect that bacterium. The likelihood that 2 strings of 11 base pairs would exactly match each other is the same likelihood is that of dealing cards one by one from a well-shuffled deck and getting 11 spades in a row. Soon he found similar matches between base pairs of other bacteria and other viruses. In fact, he discovered that all bacteria known to be resistant to infection by a particular virus always contained a set of bases that exactly matched a section of that virus's DNA. Were spacers like fingerprints or mugshots of viruses that had attacked the bacteria in the past? As you can imagine, Francisco was enormously excited by the possibilities. So that night, he went out to celebrate his discovery with colleagues and then returned to his lab the next morning, bleary-eyed, to draft a paper for submission to a medical journal. Realizing the importance of this discovery, he decided to send the paper only to the most prestigious journals. So began an 18-month odyssey of frustration. One after another, journals turned down his paper, saying the key idea was already known, or it lacked sufficient importance. Afraid of being scooped, he sent the paper to less prestigious journals. Finally, in February 2005, after having the paper out for review for 12 months, a journal finally agreed to publish it. However, by this time, CRISPR had begun to attract the attention of other researchers. For example, the giant Danish dairy firm Danisco was trying to protect the helpful bacteria that curdled cow's milk into yogurt. Too often, viruses killed them and the yogurt spoiled. A Danisco scientist, Philippe Horvath, was assigned the task of finding how to prevent these viral infections. In late 2004, he independently made the same discovery Francisco had made, namely, if a bacillus contains a series of bases in its DNA that were identical to one in an attacking virus DNA, the bacillus was resistant to attack by that virus. He also found that if a bacillus had a series of DNA bases that differed only slightly from that of the virus, it provided the bacillus with no immunity to that virus. So it appeared that the bacilli had a library of mugshots of deadly viruses, but it was not clear how they put them to use. The next step in the CRISPR mystery came a few years later, when researchers noted that near the spacers there was always a protein named Cas, and that somehow the combination of Cas and the spacers were critical to bacteria's resistance to viruses. So far, we have been exploring how the bacterial victim might work to fight off the crime of virus attacks. It's now time to examine how the perpetrator of those attacks works. A virus is made up mostly of a long strand of DNA. When viruses want to go in search of a prey, some of them roll up and travel around inside virus capsules that look like miniature spaceships. When this strange vehicle identifies a victim, such as a bacterium, it puts down its landing gear, lowers a tube into its victim, and then pumps its viral DNA into the bacterium through the tube. Once inside the bacterium, the viral DNA begins reproducing itself, using the contents of its prey as building materials. After it has reproduced enough copies of itself and its spaceships to explode the bacterial cell walls, the cell bursts and copies of the virus spread to other victims. But how do bacteria protect themselves from these marauders? Researchers looking inside bacterial cells noticed that Cas proteins were swimming around carrying segments of DNA of viruses that had attacked the bacteria in the past, just like a policeman might wander around a neighborhood carrying mug shots of known criminals. Where did these segments come from? Cas had cut them from viruses that attacked the bacterium in the past and deposited the segments into sections of the bacterium's DNA. Later, CRISPR copies them to use as a guide to aid the bacterium in quickly identifying potentially fatal viruses. Scientists named these mugshots guide molecules. They found that when Cas came across a DNA segment that didn't match the guide molecule, it moved on. But if it encountered a strip of DNA base pairs that exactly matched the guide molecule, it extended its splicers and cut the virus, rendering it ineffective. There is a hanging thread I'd like to tie up before I discuss how CRISPR can be used to improve our lives, namely, what do the palindromes do? As a bacterium adds guide molecules to its library, the library gradually grows longer and longer. When a single Cas protein wants to copy a single guide molecule to carry around as a mugshot, it needs to know exactly where guide molecules start and end. It does this by placing a palindrome at the beginning and at the end of each guide molecule, kind of like parentheses. Scientists working in laboratories began to inject laboratory animals and plants with sets of Cas plus guide molecules that exactly matched specific genes. As they anticipated, the combination sought out the target genes and deleted them. After another year or so of research, they found a way not only to delete a gene, but also how to modify it or replace it with another gene, essentially a biological word processor that could reliably find, delete, or replace existing genes with new ones. It was suddenly clear that virtually any gene in any cell of any plant or animal could be accurately targeted, then either edited, removed, or replaced. No longer was nature in sole control of how plants and animals would grow, differentiate, evolve, and reproduce. Soon, mankind would have the power to alter the basic appearance and functioning of all living things. By 2013, Chinese researchers had programmed CRISPR to find and fix a single letter among the 3 billion DNA letters of the mouse genome and thereby prevent the mouse from inheriting cataracts. Soon, scientists were making tomatoes and pears that did not rot on the shelf for months, cows that had no horns, eliminating the high cost of having a veterinarian patch them up when they were injured during transportation in trucks and trains, hypermuscular pigs and cows that produced twice as much meat, and cows that were resistant to tuberculosis. In 2015, salmon became the first genetically engineered organism approved for consumption in the United States. To produce this salmon, researchers introduced a growth hormone that reduced the time it takes for the fish to reach market size from 3 years to 18 months. Raising these salmon requires fewer resources than normal salmon, thus contributing to the production of more sustainable food products. A team of scientists in China used CRISPR to edit out 2 genes that suppress the growth of hair and muscle in goats to increase cashmere and meat production. By the end of 2015, the list of animals whose genomes had been edited included fruit flies, dogs, rabbits, and monkeys. The list of plants included rice, wheat, tomatoes, and corn, soybeans, oranges, and other plants. A mutation, as you may know, is a permanent alteration in one or more of the base pairs that make up a gene. This mutation was caused by ultraviolet rays of the sun, which are a common cause of skin cancer. Mutations range in size from a single base pair to a long segment of DNA that includes multiple genes. Working with cells taken from patient tissue samples, researchers have attempted to repair an ever-expanding number of DNA mutations associated with the most devastating genetic diseases, from hemophilia to cystic fibrosis. Sometime in the future, it may become acceptable to use CRISPR to replace faulty DNA in a human embryo. This would not only help the embryo, but once the embryo became an adult and had children, it would provide the same benefit to their children and to their children's children. Last year, scientists used CRISPR to edit the DNA of dozens of embryos that had a gene mutation that caused a serious heart condition. The embryos were only a few days old and were not implanted in a womb. Another CRISPR application may be helpful for people who need heart valve replacements. Unfortunately, there are many more people who need such replacements than there are healthy, transplantable valves in the hearts of deceased humans. Recently, pigs' DNA has been modified to make their heart valves biologically acceptable when transplanted into humans. However, no such transplants have yet been carried out. There are a number of concerns about the use of CRISPR in human beings. Some recent studies have found that the body's natural defenses may sometimes put up defenses against CRISPR. These defenses may sometimes make cells more prone to cancer. For this and other reasons, experimental treatment with CRISPR is currently restricted to individuals who already suffer from disorders known to be fatal, yet who might be cured by CRISPR. There is also the risk that CRISPR, when used to make a change in an egg, sperm, or embryo, might cause an unwanted genetic change that could be passed down through future generations. For this reason, Canada has a federal law that makes it a criminal offense with penalties up to 10 years in prison for using gene editing tools on such cells. Another concern about using CRISPR for treating human disorders is its cost. Non-CRISPR gene therapies for humans have run close to $1 million per patient, generally paid for by insurance. The companies justify this high cost, claiming that over a 10-year period, traditional therapies for certain disorders can cost 5 times more than gene treatments. In addition, they claim that CRISPR may be able to treat and cure many disorders for which there is no current treatment. A more recent concern about CRISPR is its potential for abuse. This concern has increased as it has become so easy and inexpensive to purchase the technology. What used to require years of work in a sophisticated biology laboratory can now be performed in days by high school students. I recently did a search for CRISPR online, and here is one page I saw. There are hobby CRISPR kits currently for sale online for $150 and less. There are high school workshops around the country where amateurs can learn the techniques and experiment with CRISPR. In 2015, one company shipped 60,000 CRISPR-related products to over 80 different countries. But what is the worst that could happen? My nomination would be the following. The worldwide devastating virus disease smallpox was eliminated from Earth decades ago, except for some samples of the virus that are being kept under high security by the United States and Russian governments. Recently, a research team at the University of Alberta, Canada, in just 6 months and for about $100,000, recreated an extinct relative of smallpox by stitching together fragments of mail-order DNA. To some experts, this experiment essentially rendered irrelevant a decades-long debate over whether to destroy the existing smallpox remnants, since now that scientists who want to experiment with the virus can put it together themselves. There is currently no way to prevent the intentional or unintentional development of new diseases, and there are currently few legal guidelines for its acceptable use or penalties for misuse. It is an open question whether laws can be made and enforced that will dissuade irresponsible humans from tinkering with a process that has such potentially dangerous consequences. In the United Kingdom, scientists are subject to strict oversight, but are given clear pathways for licensure by the responsible regulatory agency. They believe that this allows for careful and controlled progress with clear reporting and evaluation of results before proceeding. China has less stringent, sometimes inconsistent processes for human trials compared with their Western counterparts. In fact, in late January, China reported that it has treated 86 cancer patients with CRISPR editing. In March 2018, the U.S. Department of Agriculture announced that it will not regulate plants that have been modified through genome editing, as long as the plants could have also been developed through traditional breeding, such as crossbreeding or selecting for desirable properties. That means there will be no GMO stickers on foods developed by CRISPR. The announcement included the statement that CRISPR holds enormous promise for helping protect crops against drought and diseases while increasing nutritional value and eliminating allergens. Dr. Carl June, lead scientist from the University of Pennsylvania, has said that with regard to CRISPR, the United States is at a dangerous point in losing our lead in biomedicine. It is hard to know what the ideal is between moving quickly and making sure patients and the public are safe. In conclusion, I'd like to quote a professor at the University of California. Jennifer Doudna wrote, quote, "Let us be hopeful about CRISPR, but also let us be afraid. But above all, let us be educated. Let's not allow gene editing to run amok. But also, let's not fall prey to uninformed legislative restrictions without carefully balancing the extraordinary possibilities that CRISPR offers. The next decade will tell us how well we will be able to establish and maintain that balance."